Hi everyone, I'm Seong from University of Pennsylvania under Baraj Lab. Today I'll be talking about contrasting and combining transcriptome complexity captured by short and long RNA sequencing reads. So the goal of our project is we're generally trying to build a tool that uses both short and long reads to detect and quantify isoform in any given samples. There are a lot of long read tools that you see outside, such as all the tools that I listed. They quantify and detect transcript. But from long-read transcriptomics papers perspective, we only see what long-reads are finding, but we also they don't show what they're missing. So we're trying to explore an unbiased way of looking at both information. And to do that, we first want to understand transcriptome complexity with short and long-reads RNA sequencing. The drawing is a gene splice graph, which we can visualize complex transcriptome in a gene where the gray boxes are the exons and the drawing of the lines are the splicing variation and their types. Different splicing variation lead to different isoforms within a gene, hence understanding splicing is a very crucial step. And here we use our lab's RNA splicing tool called MUJIC to detect and quantify RNA splicing, or what we call local splicing variations, LSV. LSVs are split from a gene splice graph where you can see the splice variation coming from or into from a reference exon. And we want to detect and quantify this LSV using short and long reads, but they have different pros and cons. For short read sequencing, when I say short read, I'm generally referring to Illumina. The pros of short read is it has a high coverage and low error rate, enabling to detect and quantify splicing variation. But on the other hand, because it's short, it's difficult to identify full isoforms. For long-read sequencing, I'm referring to Oxford nanopore technology and PacBio dataset. It's good with better at detecting partial or full isoform, but because of its coverage issue and high error rate, it's generally having a hard time detecting and quantifying splicing variation. So the question is, how do we combine and contrast these two technologies in a meaningful way? So the approach is we feed short read sequencing and annotation into our RNA splicing code MUJIC and detect and quantify local splicing variation. And we feed in the matched long read data and annotation to any long read algorithms. It could be Oxford nanopore, PacBio, or all of the list of algorithms that you use. And using those two outputs on the B, we can do a lot of comparative analysis that I'm going to show in the next slides, which is mostly comparing splicing variation between two technologies or coverage difference, bias difference, etc. And then C, we combine these two outputs into a unified visualization so you can know which splice junction is from which information. It could be either short read, long read, annotation, or any combination of those. So one of these splice junctions can fall into one of the 6 categories. For example, if you look at the first row, all means this splice junction can be found in all 3 information, MUJIC, any long-read algorithm, and annotation. And for the second row, it's both de novo, which means it's not found in the annotation, but it's found in both MUJIC and long-read algorithm, and so on. But we need to handle coverage difference between short and long read because inherently they have a different coverage. So there are 2 ways to do it. The first one is to have a same cost, and the second way is to have a same base cover, and this is what we use here. When I say same base cover, when you multiply the read depths and the read lengths, you're going to get a number, and across the technology, they should be similar. And the rest of the analysis that I'll be showing, I'm using a matched short and long read human cell line from the long read consortium dataset based on cDNA, and here are the list of the read lengths and the read depths of the technology that I'm using. So I'm going to show the splice junction detection between MUJIC and long read algorithm based on PacBio and Oxford nanopore. We use 4 long read algorithms, but I'm going to show only based on an algorithm called ISOQuant, not on the other algorithms, because ISOQuant can be used and ran with one command option, which was really easy to finish the analysis in time. And I have all the other analysis based, but I can show you that after the presentation because those are not in the presentations. So if you focus on the left top, this is a splice junction comparison between MUJIC and ISOQuant based on PacBio. If you look at the blue box on the left side, you can see approximately 66% are found by MUJIC, ISOQuant based on PacBio and annotation. And if you combine the green one and the red one, approximately 22% are only found by MUJIC. And if you combine the pink one and the yellow one, approximately 11% are only from my long read. And similar for if you look at the right side, we're comparing MUJIC and ISOQuant based on Oxford nanopore. So you can generally see there are difference between different splice junction detection when you use ISMB or PacBio. But now we want to know if these junctions are actually meaningful. So to do that, we took the number of splice junction detected by MUJIC, if you look at the left bottom part, and assess how many isochron PacBio or Oxford nanopore recovered from those in the function of psi. When I say psi is a short for person splice in or junction usage. When the psi number is close to 1, it means junction is used more. When it's close to 0, it's used less. So if you look at the left bottom again, you can generally see long read is suffering from the low psi values, because when you compare the red and blue color with the gray one, you can see generally they're very low. And if you look at the right side, this is a cumulative distribution function of the left plot. And you can see PacBio misses approximately 10% junction compared to what MUJIC finds, and Oxford nanopore misses 30% of the junctions that MUJIC finds. So the question is, are those junctions even real? For a short read, likely yes, because for a short read junction, they are supported by multiple unique reads that map distinctly to multiple position. And for when we use MUJIC, it has a conservative detection threshold, such as the annotated junctions needs at least 3 reads, or de novo junction needs at least 5 reads, etc. For a long read, likely not, due to a higher rate and limited coverage. So we want to know the huge discrepancy between these two technology. And from the rest of the slides, I'll be showing the main 3 reasons that we found. So the first one is the coverage difference. So similarly with the previous plot, we took the number of splice junctions deducted by MUJIC and assessed how many long read can actually recover in the function of number of reads. And if you look at it, and generally, long read can have hard time recovering when the reads are really low on the left one. And if you look at the right side, when I circle the one that I circle with the red circle, you can see PacBio misses approximately 11% and Oxford nanopore misses 18%, even though the reads are more than 100 reads. And again, when we compute this in a CDF plot, you can see PacBio finds approximately 70% of the total number of junction that MUJIC finds, and Oxford nanopores finds about 52% of the junction that MUJIC finds, which is pretty, you can see the huge discrepancy. So we can see that covers this difference actually lead to a difference between the two technologies. And number 2 is could be due to 3' to 5' bias. So we explore how many long reads cannot quantify LSV that MUJIC can quantify based on distance from 3'N. When I say quantify, you need an LSV of at least 10 reads. So if you look at the black circle that I had, this is Oxford nanopore, approximately 68% of LSV are not quantifiable when LSV are more than 2,500 base pair away from the 3'N. But still, if you look at the orange one, about 38% of LSV are still not quantifiable when LSV are very close to 3'N. And if you look at the red line, PacBio has a generally low non-quantifiability, and you can see the non-quantifiability is flat compared to Oxford nanopore, but you can still see a lot of non-quantifiability across the two technologies. And this is a figure, this is a plot that, this is a read length distribution we plotted using the long read consortium data set. And if you look at the gray line, which is on the left side, this is the Illumina. Illumina has a very constant read median length, and you can see the read density is very high. Unlike Oxford nanopore and PacBio have a very different read length distribution, and the read density are very low compared to Illumina. And only 5% and of Oxford nanopore and 50% of PacBio from this data set spend 3,000 base pair or more, which is human transcript length. So you can see long read actually don't spend the human transcript that much. Another reason we also discover is GC content around splice junction may help explain their differential detection by short and long read. So to explore this, we selected junctions that are more than 40 reads and computed the GC content around the splice site of 100 base pair, so left to 50 or right to 50, on the function of distance from 3'N, similar with the previous plot. And we divided these into 2 groups. One is the red and the blue one is, one group is the junction that are found by both short and long read, and the green ones are the ones that only short read can find, but the long read misses. So if you look at the 2 plots, you can see the gray one has a higher GC content compared to the red one or the blue one, which means the differential between long and short read can be explained by the GC content proportion. So to see if GC content is actually affecting like a local structure, we computed the y-axis using MFE, medium and free energy, using RNA-fold. So you can see the gray one has a lower MFE, which means the junction that short read only finds and long read misses are likely to have a more local structure. So local structure associated with higher GC content and low MFE may be a contributing factor that can explain difference between short and long read. So these are the three main factors that we find why long and short read can be different. But so far, I've been only investigating junction, but what about interim retention? The left, focus on the left one. So left plot is an offset plot of showing interim retention between Mujik and ISMB. And if you look at it, ISMB based on PacBio finds about 10K of interim retention, which is a lot compared to ISMB Oxford nanopore, which is 6.3, and Mujik only finds about 2.4K. So in general, you can see long read finds a lot of interim retention due to its longer reads. But if you look at the right side, this is a interim retention length comparison using the left offset plot. So looking at the left one, you can see that Illumina detects longer interim retention. So short read generally detects longer interim retention compared to long read. Because if you look at the rest of the other box plots, they include long read and the length of the interim retention distribution is much shorter compared to Illumina. So finally, I want to introduce our unified visualization FOLA based on who San made it. So for people who don't know what FOLA is a visualization package within Mujik, and it was only supported by short read. And for this example, if you look at the first row, the gray boxes are the exon, the red lines are the junctions and that are supported by shortened annotation, and the green ones are de novo ones, and the dotted gray lines are only one that supported by annotation. And if you look at the bottom one, you can see the psi per junction per each LSV type. So this is the original FOLADA if you're used to it or if you're used to it. But now we extend it to long read transcripts. So you can also visualize long read transcript and see like all the junction per read and the exon reads. And you can also compute long read psi per junction and each LSV type. But what we're most interested in is we want to combine these two CNs, see a unified visualization. So the second row is what we came up with, and we call it, again, FOLA. So this unified visualization is based on Mujik and ISMB PacBio. If you look at the second row, the color matches with the bar plot that I showed previously. And for this example, this gene is an SRS11, which is a pre-mRNA splicing factor. And the black boxes is known to have an ultra-conserved PTC that can trigger NMD. But if you look at the orange boxes, you can see long read captures an extra interim retention event that short read misses, and it can also find an associated full isoform. So with this unified visualization, you can see which technology is missing and finding, and you can use it on your own needs. So for summary, we built a general framework to assess long read technology algorithms by comparing to short read. And long read is the future, but currently there's a lot of things missing depending on your need and usage. And so far we found the main reason there are differences because of coverage issue, 3' to 5' bias, or local structure associated with GC content. So if you want to do your experiment, you can choose your technology and tool based on your needs. If you want to do discovery, probably short read. If you want to focus on isoform, do long read. So we built our tool called Magic L, where we call it magical, to combine short and long reads in a unified splicing analysis to see any genes or LSB you're interested in. You can see what its technology it's finding, where they agree or disagree, or you might be missing with either. So for last, I want to thanks to my lab and our funding, especially to my PI Yose, collaborator Andre, co-author San. And this is the end of my presentation. I'm happy to answer any questions. Thank you.
Thank you very much. Okay, so we have a lot of time for questions. Are there any? Yes?
Hi, thank you so much for your presentation. I love this integrated visualization of the long and short reads. So you have been using the longer gas data that already has some time. Now we are seeing, for example, PacBio data sets with 40 million reads in which we have many more junctions detected that with regular Illumina sequencing. So how do you think that your framework may change the conclusions when you are having not that much of a problem of coverage as you are reporting here?
That's a great question. So the reason why I choose more than one reason, so I basically talk about their coverage, 3' to 5' bias, and like GC content issues. So for PacBio, if you have more coverage, I assume that the coverage is better, and obviously it's gonna have if it's gonna have less 3' to 5' bias because it's longer. PacBio is longer than Oxford nanopore. So I do think it's gonna solve the problem a lot. But for our project, we're not trying to solve like long read issue like because we all use short read and long read, and via any other data set, you can always like combine these two and have like different splicing analysis. So for your question, it will find more splice junction, but if you're the user, I hope everybody uses it and see how it looks like. And I can't answer like how GC content is gonna affect 3' to 5' or coverage, because all these three are gonna combine and affect the difference between short and long reads.
Thank you.
Any other questions? I actually have one. So you've seen the dependence on GC content, but did you also look at how the GC content influences the Illumina coverage? Maybe if there is some interplay between that.
So the question is, if I look at GC content at Illumina, is that the question, if I understand correctly? So if I clarify what I showed here, yes. So in here, when I
We don't see it anymore.
Oh, sorry. Yeah, I got it.
Okay.
So when I say we took more than 40 reads, it means we took all the short reads that are more than 40 reads supported. So we basically use the reads based on short reads, because long read can't find all the junctions that short read can find. That's why we extracted the reads from short read that are more than 40 reads and computer difference. So we actually used it based on Illumina, if that was your question.
And also with short reads, sometimes you cannot map them uniquely. Did you look at that? Like whether the long reads sometimes can be easier to map to the unique location?
Are you talking about the mapping such as Minimap or like star?
Well, doesn't matter. Like if there is like a gene that's duplicated and part of it it's identical, you wouldn't be able to map the short read uniquely to one of the copies of the gene, but the long read maybe might be.
I see. That's a good question. No, we didn't look at a specific example like that, but hopefully we can try to look at it with our new tool, Magical.
Okay, thank you. Any other questions?
Thank you for a nice talk. So I just wanted to ask about if you could elaborate more on the data.
So can you speak louder, please?
Yeah, so the data was not very clear, especially the long read. I was a bit surprised about the quality, the ONT, like, seemed to be very short. Like, is it?
So it's the question about if the long read is actually short.
So the long read, I think the long read data seems quite like short or bad quality. So the PacBio was already discussed. Now with Kinect, you get much better throughput and quality. And the ONT, I don't know what kind of data is it.
Oxford nanopore usually, so in this project, we're using RNA and not DNA. So RNA, DNA, long read have a different read lengths. And for this example, we're using a public data set called long read consortium data set. And if you're asking about the read and the coverage in general, for Oxford nanopore in RNA, it's usually spent between 600 to 800, and for PacBio, it's about 2,000-ish to like 3,000-ish. So for the presentation I gave is based on long read consortium data set, and those are the read lengths, and that's the answer for your question. But we also try with GTEC and also cell line from BAL data set, which is not part of the presentation. But if you want to know more, we can talk more after the presentation.
So maybe I can answer this question because I generated this. I was in the consortium generating the nanopore data. So yeah, that's surprising, but that's a fact that when you are doing cDNA and even direct RNA sequencing with nanopore, the read length is short. And that's a limitation of the technology. It's very different with DNA, but with cDNA, that's what you get.
If I add another one with that question, is for Oxford nanopore, it could be generally shorter, but compared to PacBio and RNA sequencing, Oxford nanopore generally have more coverage compared to PacBio. And the Oxford nanopore can also do direct RNA sequencing, which means it has, because you're not converting to cDNA, it has less 3' to 5' bias. But maybe the coverage is smaller than short read, or still there could be like GC content or local structure bothering the long read because long read only starts from 3 pi men. So that's why we're trying to look for more than one reason and trying to see a lot of reasons that can complicate the problem between short and long reads.